not here. I think actually everybody's here now. But that way I can mess with it, or I can at least have it so you can go back and look when you get confused. So if I was going to take notes, I would call these genetic notes. And this is what we call Mendelian genetics, is what we're going to concentrate on first, or kind of Mendelian. And if you're playing along, I think this is 5.2 to 5.4, something like that. I divide it up into something that's pretty much the standard stuff you've been taught. I call that Mendelian. It's not 100% true. And then I call it non-Mendelian. But there's a couple of terms. And then what I'm going to do is just work some problems. I'm just going to go over some problems. And then I'll give you the worksheet. The worksheet's not due next class. I'm going to give you the worksheet to kind of work on so that when you come back to next class, I can work problems for you that you have issues with. We have to make sure that the foundation that hopefully got placed, in 7th and 9th grade stayed, and so that you can think and do, so that you don't get frustrated when Punnett square goes away, and then we start talking about wild types and mutant types, and we change the system that you use. So we need to get the basic piece done first. I don't want to make an error and make an assumption that you know how to do it. These two pieces, Mendel's law of segregation, Mendel's law of independent assortment, that is concepts you have to know and are tested on. Typically, what I saw, the test questions were multiple choice. Okay, multiple choice. So they might give you a scenario and ask you which one it is, or they might put, which of the following statements, let's see, what does it say? Which of the following statements is correct for Mendel's law of segregation? And then they had these statements there, and you have to pick out the right one. Okay? So these two. The law of segregation basically says that parents pass on one gene. Remember, everything is paired, right? Parents pass on one gene. That's pretty much it. There's a little more to it than that. So parents pass on one gene. So remember, you got to remember that the parents are diploid, right? And what they do is they create through meiosis that haploid sex cell, and so one of their pair of gene gets passed on. That's what that says. Law of segregation. The diploid segregates out to haploid. That's what this is about. So far, so good? And then the law of independent assortment basically says that as a parent, I don't sit there and go, okay, I want this gene to go here, and that it's all random. How they separate out is random. And the way they word it in AP and in IB is independent, make sure I write this correctly, independent from one another. So I want you to think random here. So one parent, or a parent gives one gene, and what gene they give of the pair is randomly given away, is randomly put. There's no conscious effort on that. Now, I have a problem with this definition, and here's where I have that problem with how we do this. It's not so much one gene as it is a chromosome. And here's the reason why, and this is going to be more later. We're going to hit this more a little later in the genetics piece here. But you had chromosomes on the thing we just finished, and it had like a big G and a big L, right? What this says is that the whole chromosome moves, not that this splits up and moves, but the whole chromosome moves. So all the genes that are on one chromosome, because you have two chromosomes, right? So if I had two of these chromosomes, let's call this little g and little l, all of those genes are going to go together on that one chromosome. That's what the segregation is. And that which one of these goes, which one of these makes it into that sperm is random. Do you follow why I have a problem with the gene? Because that's a gene and that's a gene. Do you understand why I have a problem with using the word gene?
Yes.
You guys okay? Okay. This is a concept that they will ask you questions. If you understand the concept, you'll get the question every time because it's mostly multiple choice. In fact, I don't recall seeing any question that was a short answer on this, that you had to explain it. It was all, here's a scenario, or here, which one of these statements is best for that. Is there any questions about this at all? Okay? Mom and dad give one copy of their genes to the parent, to the offspring, and that copy that's picked is randomly done. They're not making a choice. Any questions about that idea? We good? All right, can I erase, or do you need to take a picture, or do I need to wait, give you a minute? All right, let's work some genetics problems here real quick. Alrighty. This will be fun. Now, you remember Punnett squares, right?
Yes.
Yeah. All of Punnett square is a way of doing a probability statement. Now, this is not, it is probability like what you get in statistics from, what is it, Babb and Schwartz? Are those the two statistics teachers, I think? Those are the two guys that do statistics. I think there's only two. So it is like that, but we just kind of look at it a little differently. A little different than what you are probably accustomed to. Grab this so I make sure I don't hit things kind of weird. Alrighty. I'm going to start off with something straightforward. If I were to have this cross, some gene A, the big A and the little a, those are the alleles. Everybody okay with that? I'm making an assumption you know that. And that the big A is the dominant allele. And the little a is the recessive allele. So far, so good? You remember all this. What would be my percent offspring that are little a, little a? I don't want you to write anything down. I want you to look and see, if you can do this in your head, what percent of the offspring, probable percentage, would we have to have little a and little a offspring? 25%, yeah. Did you need to do that in your brain? I mean, excuse me, on paper, sorry. No. You didn't need to do that on your paper. You need to do it in your brain. Because we know these guys go together. So far, so good? What if I had it like this? Would I get any little a, little a? No. What percent offspring would be big A, big A?
50%.
Okay. I hear 50%. I heard 75. 50%, is that what you want to go with? I heard more of that. All right, so let's look. Because of segregation, because of independent assortment, that means this big A would go with that big A. That would happen, right? This big A would go with that little b, that, or little a, excuse me, that would happen, correct? The same thing with the other one, that big A would go with that one, so we'd have another one of those. And the same thing here, so it would be 50%. I literally just did the Punnett square without writing everything out. So far, so good? Okay. We need to talk a little bit about what dominant is and what recessive is. It is now time for us not to lie to you and tell you that dominant, big A, gets you the phenotype. It does, but dominant means expression of the gene. Dominant means that if I had a chromosome that had, here's my diploid number, right? I had a chromosome that had big A and little a. It means that this one will be copied. This one will make the protein. This one will not. This one may get expressed, but there's something that it's not going to work. But it's usually this one that gets copied. Dominant means expression. And expression of the gene means that the protein is being made. But that protein may fit into a larger piece to actually get the phenotype for hair color, or the phenotype for skin color, or the phenotype for height, or the phenotype for being able to put your thumb back that I can't do because I'm a mutant. I'm totally recessive when it comes to this. You guys can all have hitchhiker's thumb. Almost every one of you in here does. And by the way, more scary thought is if yours doesn't go back, that means you're more related to me than you are to anybody else in the room. Okay? Recessive means no expression or limited expression. Or there's something wrong and it's not, it's being expressed, but the expression doesn't cause the phenotype to occur. So you come out of ninth grade, a lot of times you think, oh man, dominant. That means it's stronger. No, we need to tie it back to the actual gene. We need to tie it back to an actual gene. And I'm not ready to go over this yet, but I want to be sure that you start thinking about this process to get to our product at the end. Our DNA has our genes, and that is then put onto mRNA. That's our copy, which then goes to a ribosome to be made. And that ribosome then makes that protein. And all of these things have a code in them. This is the flow of information, the biological flow of information. Your parents gave you the information and you were created. Your grandparents gave the information to your parents who then gave that to you and you were made. And it goes through this process using meiosis, transcription and translation, transcription and translation. You guys follow? And we'll talk more about this concept when we hit translation and transcription. Right now we're dealing with the flow of the genes, the information between parents. Are we good? Okay. All righty. So let me show you some tricks. I'm going to erase. And again, we'll hit this again. It's actually called the central dogma. It is the one central idea of all of biology. But we'll get to that later, so don't panic. Okay. All right, so let's do some more of the genetic stuff. What would be all the possible offspring produced? And I want the genotype, because I'm not giving you phenotypes yet. I want the genotype of all possible offspring produced. How many?
2 types.
2 types. Let me show you a trick to figure out how many types there would be produced. We can just look at this right here, right? this person, what kind of sperm would it have for A? It'd have big A sperm. In fact, that's the only A thing it can make, right? So there's only one opportunity here. So big A and big A, right? And I'm going to do it a little overboard here, so just kind of watch at the moment. All righty. For this person, what type of sperm can it make here? B. So we could make big B, right? Actually, you know what? I'm going to skip a step here because I think you guys can handle it. So far, so good? This person here, what are the possible gametes it can make between big A and big B gene? The A and the B gene. What are the possible gametes it can make? So far, so good? I know it can make 4 gametes because there's 2 options here and there's 2 options there. So that means I can only make 4 different types of gametes, right? I can get 4. But of those 4, how many of the little a or big A, little b do I get? So far, so good? Sorry, I'm in a little bit of a hurry. So do we have it right? Okay, I think I shouldn't have skipped that step now that I see this. Let's do another one of these things. I'm going to stop this and we're going to do another one of these things. How many different kinds of gametes can that individual make? All right, how many possibilities are for big C, little c? Big D, little d? Little e. Because see, this is just going to be E and G, right? Right? So you're either going to have a big C and a big D that goes with that, a big C and a little d, a little c and a big D, a little c and a little d. 4 different types. Okay, we good so far? I'm going to erase. All righty. So that means, I'm going to come back to here. So that means here, how many types of gametes can this make? Make 1. What's the gamete that it can make? That's the only one it can make. So far, so good? All right, here, how many types of gametes can this one make? 4. What are they? Oops, I just wrote that again. Thank you. Right? So that means that this gamete could go with that one. That's a possibility, right? It's also a possibility that this one could go. It's also a possibility that one could go, and the possibility that one can go. What percentage of time do I make big A, big B? What percent would that happen? 25%, right? What about this one? What about this one? What about this one? So that means that if I had, what percent would this one be? 100%. That means that I've got 100% here, so basically, what percentage of time would I get this? 25%. And this one would be big A, big A, big B, big B. So far, so good? For here, it would still be just 25%, right? Because it's 25% of 100, which is 25. That would be big A, little a, big B, big B. This one would be big A, little, let me put it in the right spot. Again, 25%. So far, so good? Any questions about that? You can still do a Punnett square, but your Punnett square, I actually had a teacher tell me that they made the kid do this, and I was like, what is wrong with you? They actually made the kid write oops, I don't have enough on the end here. They actually made the kid write that 4 times. Absolutely ridiculous to do that. Absolutely ridiculous. Because you only have the one, right? So you could still do the Punnett square here. We just need to get you past that. Because remember, it is a part of the whole. It's just a percentage. So far, so good? Okay. I'm going to erase. What are we looking at on time-wise? Oh, good. We're good. All righty. So on the worksheet, there's a thing that I realize that you guys don't quite know yet. Let's just do it this way. Let's say I have this as a genotype. And let's say big A is red. And it is completely dominant to little a, which is white. So far, so good? What's the phenotype of that? Red. Does it matter what goes here? Does it matter what goes in that box? The phenotype is still going to be red. If you're looking at genotype, it might matter. So far, so good? So if I had a problem that looked like this, and I had one offspring that was little a, could you tell me what that one was? Okay. If it was this, what would go here? Big A? It could be either, right? It could be either. Sometimes you'll get things that are written like this, and it'll say this is red, and then it'll say this is white. What does that underline mean? It means it could be big A or little a. When you answer the question, you have to give the full phenotype. Don't just put a line there. You guys got it? Or genotype, excuse me. You guys got it? All right. Type A blood. This means on their cells they have A antigens on the outside of their red blood cells. Does anybody remember what the genotype for A type blood is? Okay. And recently, because of the way it is on the STAR test, we've had teachers teach the wrong thing here. There are only 3 antigens. 3 antigens. And there is an A antigen, a B antigen, and an O antigen. An A antigen means you're presenting A antigen, you're presenting A, The body's seeing A, that's you. B is B. O means you don't have either one of those. So for us to do this multiple allele, see, we have 3 different alleles that it could be. We have a little different system that has these 3 antigens. Type A blood, you have to use a capital I. The capital I stands for immunoglobulin. It tells everyone that that's what you're using. And you superscript the allele. So type A blood could be that. Big I, big A, big I, big A. But because A is dominant, see, this is a lowercase letter. Because A is dominant, you could also have, and you have to use this. If you put it in any other format, wrong. There is no blood type that is this. I do not care what the STAR test says. I don't care what your other teachers say. That does not exist. That is wrong. You will get it marked wrong. Okay, so for B, what do you think the genotype would be for B? That would be 1. IB, IB. What about type O? I, yep. Little I. And then AB. Just because they are like this doesn't mean you can't do a cross. You just need to consider all of that the allele. Do you guys remember this? I'm sorry if you had a teacher that taught you wrong. This is the correct way to do it. It is the only way that that AP will accept. Okay? I'm almost done here. So far, so good? So just out of curiosity, can we get any type O blood here? No. Can we get type A?
Yeah.
Can we get type B?
Yeah.
Can we get type AB?
No.
Okay? What gametes would this one make? What gametes would it make? No, this wouldn't make, there's no little I here. What gametes would this one make? That's it, right? What gametes would this one make? Little I. So basically, you're going to get 50-50. So far, so good? Okay. Sex-linked. I think I have enough room. Also sometimes referred to as X-linked, or rarely, I don't really see it with y'all, I see it with the IB, you have Y-linked. This means that the chromosome, that the gene is on the X or the Y chromosome. Okay, we'll talk about what actual linked genes are later. Classic example is male pattern baldness. Gentlemen, you need to look at your mom or your grandma. If your grandma has thinning hair, chances are you will too. So a female could have, I'm going to use B, could be homozygous, dominant. We write it as a superscript because we're saying that this gene is located on the X chromosome. Okay? It doesn't matter what dad has, so dad could be this, but all the boys are going to have the dominant B. So far, so good? You guys see it? You have to write it this way. Has to be written this way when it's X-linked. Okay? If I had a female that was heterozygote, what would be the chances of getting a male that is colorblind? I'm not saying the chances of getting colorblind males, I'm just saying a male that is colorblind. All right, well, let's look at it. Erase this over here. Let's look at it. You have to watch the way the things are worded. The chance of a male with balding, male pattern balding. So here, what would we get for our genotype? X big B and X little b. Here, what would we get? XB. No, genotype would just be XB. And, just a second. Okay, so I'm going to go ahead and do a Punnett square because I see some curious looks in the faces and maybe I'm reading it wrong. X big B, X little b, X big B, Y. Surprised I still have it. What percent, listen to the question, what percent of the males would be colorblind?
Because it only counts for the Y ones.
That's right, so it'd be 50%. What if the question asked, what percent are males that are colorblind? Still be 50%, right? What percent of the offspring are not colorblind, balding? Sorry, I said colorblind. 50%. Watch the way the question's worded when you answer the question. Yes, ma'am.
I think this is probably a dumb question, but in regards to percentages, we know that for 25, 50, 75, whatever.
Right.
If we have more than that, how are we able to calculate the percentage?
So let's say that we had this. I'm making stuff up, okay? It doesn't fit this thing, but let's just say I did this. Okay, so now we have 2 of these out of a possible 6. That's how you would do that. Does that make sense?
Okay.
Okay? There is another little trick, but I think after the beginning, I don't want to throw that other trick at you yet. But there's another trick where you can multiply out the probabilities to do it. Any questions? You guys got it? Okay, here's what I want to do. Past experience has told me to give you guys the worksheet, let you work on it, tell you it's not due next time, let you work on this worksheet, and then when you come back, see what problems they are, answer those problems, and then we move on from there.
Sounds good.
Sounds good? Okay, that's it. Let me get you the worksheet.